Okay, good afternoon. My name is Kozo Kaebuchi from Nagoya University. I'm going to chair this symposium. Also, it's a luncheon seminar. So today's speaker is Dr. Adam Pawson, who comes from Edinburgh. He's in charge of the database, a famous database in the pharmacology, Guide to Pharmacology. So I think, as far as I know, that this is the first time for us to listen to this kind of a lecture from Guide to Pharmacology. So he came to Nagoya yesterday, and he said he didn't sleep well, but he's going to give a talk about how to utilize Guide to Pharmacology. So please enjoy this. So Dr. Adam Pawson, please.
It's a huge privilege to be at this meeting, and I would like to extend my sincere thanks and appreciation to the President of the Japanese Pharmacological Society and the meeting organizers for allowing me to be here to present this seminar to tell you about the IUPHAR BPS Guide to Pharmacology, or GtoPdb as we call it. And it's our hope, the database team based in Edinburgh, it's our hope that that the members of the Japanese Pharmacological Society, along with their colleagues and postdocs and students, during the course of this meeting, will find it to be a useful tool. Okay, I have no conflicts of interest to disclose. Right, so just by way of an overview of the talk this afternoon, I'm going to start off, which I think is quite necessary, to give you a background and a history of the database, and also how we came about constructing the Guide to Pharmacology database. I'll also then introduce the committee, the IUPHAR committee that oversees the development of the database. That's NC-IUPHAR, or the IUPHAR Committee on Receptor Nomenclature and Drug Classification. I'll then go over some details on the content of the database. I'll then take you through how you can navigate the website and use the search tools. Discuss some recent additions, just with the recent targets that we've added to the database in the last few months. And then I'd also like to tell you about a publication which is now produced directly from the database called the Concise Guide to Pharmacology. And then I'll go through some additional features and resources of the database. Right, so back in early 2011, a collaboration was initiated between IUPHAR and the British Pharmacological Society. And the aim really was to develop a web portal to pharmacological information on drug targets and their ligands. And so the concept of the IUPHAR BPS Guide to Pharmacology was developed. And really, the Guide to Pharmacology, the idea behind it was that it would be a single entry point to pharmacological information that was originally contained in two fairly well-established resources, the one being the IUPHAR database of receptors and ion channels, which many of you might have known about before, and that was the original database of IUPHAR, and the other was a series of publications by the British Pharmacological Society called the Guide to Receptors and Channels, or GRAC. So it wasn't a database, GRAC wasn't a database, it was just simply a series of publications. So just a brief history about the IUPHAR database. IUPHAR-DB has been in development at the University of Edinburgh in Scotland since about the year 2000, and its development was guided by the IUPHAR Committee on Receptor Nomenclature and Drug Classification, or NC-IUPHAR, as well as an international network of expert subcommittees, including well over 700 scientists. And IUPHAR-DB provides an in-depth coverage of the properties and the pharmacology of G protein-coupled receptors, voltage and ligand-gated ion channels, and nuclear hormone receptors. So those were the three target classes that were included in the IUPHAR-DB. Quite differently, GRAC, or the Guide to Receptors and Channels, as I said, was a series of publications which was published once every 2 years as a supplement of the British Journal of Pharmacology since 2004. And where GRAC differs from IUPHAR-DB is that it provides a rapid overview of the key properties of a much wider range of drug targets or potential pharmacological targets. And I'll give you some more information about that in a second. And importantly, the difference about GRAC is that the information is arranged very succinctly so that a newcomer to a particular field can identify the most important elements of a particular drug target at a glance. So this is the front cover of the GRAC publications, and it was widely distributed at conferences in the UK and in the States. And this is a typical target family page on GRAC. So this is for the histamine G protein coupled receptors, and you can see that it included a very brief overview at the top, a table with pharmacological information for the 4 histamine receptors, some comments about the data in the table, some useful reviews to refer to, and then references. And if I just zoom in on the table there, you can see that the 4 receptors are arranged across the table. There's information about the gene, the transduction, then the key ligands that can be used to study this receptor, so selective agonists, antagonists, and radioactive probes that you can use to study these targets in the laboratory. So what we had to do in terms of constructing the guide to pharmacology was the first step, which took well over a year, was that our team of curators manually curated all the data from the GRAC publication, including all the ligands and their activity data and all the references, into a relational database. So if you take a look at the histamine page over there, and, for example, look at the H4 receptor, this is what the histamine page in the new database looked like. And so you can see it's very similar. You've got the overview, the 4 receptors, the comments, the further reading, and the references. And if you just compare that to the H4 receptor in the original GRAC, you can see that it's fairly similar, although we have added some additional features. So, for example, we now link to the human, mouse, and rat genome databases, the ensemble for human, mouse, and rat, UniProt entries for the human, mouse, and rat receptor, and then the ligands. And the difference here is that these are all links through to those particular resources, and also the ligands. Each ligand now has its own ligand page, including structure, and all the references are linked directly to the citations in PubMed. And I'll go into some more detail about that in a second. So, once we had created once we had converted all the data in the GRAC publication into a database, we then integrated it with all the data in the original IUPHAR database. And then that resulted in the production of the IUPHAR BPS Guide to Pharmacology. So, Guide to Pharmacology database has a number of aims, and I've listed them here. I don't expect you to read them all, but principally to provide access to the data on all known biological targets, and also we make recommendations for ligands to use to characterize these targets, an entry point into pharmacological literature, educational resources, and the development of the Guide to Pharmacology database is still overseen by the Nomenclature Committee of IUPHAR. So I'll just briefly tell you a little bit about NC-IUPHAR. NC-IUPHAR has a number of objectives, principally, and this is why it was originally established a number of years ago, is to issue guidelines on the nomenclature and the classification of human biological targets. It has a number of other roles, including the development of the Guide to Pharmacology, and they publish a number of very highly cited review articles in pharmacological reviews, as well as articles and editorials in the British Journal of Pharmacology on the state of the field of various target families. And this is the core committee of NC-IUPHAR, which is was chaired by Michael Spedding, and you see Professor Kayabuchi and Professor Yoshikatsu Kanai are members of the core committee of NC-IUPHAR. NC-IUPHAR also has a number of corresponding members which increase the global representation of NC-IUPHAR. And then I mentioned the NC-IUPHAR subcommittees. These are all the chairs for the subcommittees. So we have more than 90 subcommittees comprising more than 700 scientists. Each of these subcommittees is for a particular family of, for example, G protein coupled receptors. Here are the ion channels, the nuclear hormone receptors, and then various other target classes. And I'd just like to stress that the makeup of these subcommittees and the membership of these subcommittees is constantly changing. For example, we appreciate that this is all voluntary effort on behalf of these people, and people are very busy doing their normal jobs, so they can't always devote as much time as they'd like to this. So we're always looking for new experts to help us in keeping the database as up-to-date as possible. Right, so what about the content of the Guide to Pharmacology database? So if you look at the targets, we include annotation on more than 2,700 established or potential drug targets and related proteins. So these include the GPCRs, the ion channels, nuclear hormone receptors, catalytic receptors, kinases, proteases, other enzymes and transporters. And that graphic just gives you an idea of the numbers over there. Clearly enzymes is the largest number there, although the annotation of the enzymes is still very much in development. And then as far as ligands in the Guide to Pharmacology is concerned, we include the annotation on more than 7,200 ligands and drugs, including approved drugs, synthetic organics, metabolites, hormones and neurotransmitters, natural products, peptides, inorganics, antibodies, and then labeled ligands as well. Right, so navigating the Guide to Pharmacology, how do you get around the website? Well, just like many databases and websites, there are a number of ways you can do this. Principally, you can start by browsing through lists of ligands and targets, or you can use search tools. You can search by name, keywords, identifiers, or in the case of ligands, you can actually even search through ligand structures or substructures. Target families are listed under expandable family trees, which makes it a lot easier to navigate through them. And we provide information on targets at two levels of detail. The first being a concise family overview, and secondly, a detailed pages for selected targets. And every single ligand in the database, as I said, has its own ligand page. So to start with the first level of annotation, that's the concise target family. This is essentially the level of detail that was contained in the original GRAC, or the Guide to Receptor and Channel publication. So it provides, it introduces the families, the main properties of the families. There's expert overviews and comments with recommendations on selective ligands that can be used in the laboratory, clinically used drugs, endogenous ligands and probes, and then also further reading lists. As far as the detailed pages are concerned, those are the pages that were originally contained in the IUPHAR database, and there's a lot more information you can see presented for these. And all these data are collected and reviewed by the NC-IUPHAR subcommittees and individual experts where a subcommittee has not been formed. And you can see there's a lot of information contained there, for example, gene and protein information, all the pharmacology, signal transduction mechanisms and tissue distribution, functional assays and physiological functions, mutations and pathophysiology and biologically significant variants, and a whole lot more. I haven't got it all listed there. Other features of navigating through the website is that all the data is extensively referenced and linked to all the primary citations in PubMed. The focus is on human data, but where there are species differences, we do provide information on other species. All our data is extensively linked to corresponding entries in other resources such as UniProt, EntreGene, OMIM, Chembl, PubChem, and ligand information includes 2D ligand structures, peptide sequences in the case of peptides. We provide clinical data, and again, all our ligands are linked up to corresponding entries in a number of other resources. So this is the home page for the IUPHAR BPS Guide to Pharmacology, which can be accessed at www.guidetopharmacology.org. And the first thing I'd like to point out is that the navigation bar across the top over here remains on the database page as you work through the website. So you can always navigate through the website no matter what page you are through this navigation panel at the top. So you can browse through lists of targets and ligands. There are a number of resources which I'll go into a bit later. There's a quick search function at the top here, an advanced search function over here. Alternatively, from the front page, you can also directly start browsing through lists of targets and ligands through these two boxes over here. The front page also provides information on the latest release of the database. This is actually the previous release. We actually released a new version on last Friday. We also provide a news section, so information, for example, hot topics, important papers that have just been published, or in the case of orphan G protein coupled receptors, new receptor ligand pairings. And then further down the page, there's additional resources such as social media and that sort of thing. So if I navigate to the G protein coupled receptor landing page, this is the top half of the list over here for all the G protein coupled receptors. And I've chosen today just to stick with the histamine receptors just for conformity. So then you navigate to the histamine family page. So this is the family summary page for the histamine receptors. So you've seen this page already. There's the overview of the 4 receptors, comments, and further reading. So if I go to the click on the Show Summary tab for the H2 receptor, here we go. This is the information for the H2 receptor in the Concise view. So links to the gene pages, ensemble, UniProt, the individual ligand pages with affinities, and the references where those affinities have come from. So if I click on the reference number, this brings up the reference, which then links out to the citation in PubMed, where you can go and download the paper and read about it some more if you so wish. From the Concise summary page, you can also navigate to the more detailed page for the H2 receptor. And this is what the top of the H2 receptor detail page looks like. And there's a table of contents which you can click on to take you further down into the detailed view page. So here you can see this is all the information that's provided for the H2 receptor, starting with gene and protein information, and then synonyms or previous or unofficial names used for this receptor. And then this is an extensive list of link outs to other databases where you can get a vast amount of information about this receptor. And then here we start with the actual pharmacology of ligands at this receptor. So we provide information on species specific selectivity for ligands at this receptor, information on endogenous ligands, where ligands are labeled, this is indicated, and those labeled ligands that are radioactive is indicated. We also indicate which of the ligands are approved drugs, and we also indicate where this is the primary target for a particular ligand. And if I then click on the ligand name, for example, in this case, ranitidine, this brings up the ligand page for ranitidine. So if I just zoom in over there, you can see there's the 2D structure. And you can actually click on that structure, and it'll take you through to another page where you can actually edit the structure and then search for similar structures in the database using various criteria. We also provide calculated physical chemical properties for the ligand. So this is the summary tab for this ligand. There are a number of other tabs across the top there, which I'll get to in a second. And this gives you the class of the compound, whether it's an approved drug, and where we got that information from. So in this case, yes, from the FDA 1977, the IUPAC name, international non-proprietary name and number, and then synonyms. And then, as I mentioned earlier, there's also link outs to other chemical databases where you can get more information about this ligand. if I click on the biological activity tab, this gives you the activity of this particular ligand across targets in the database. So in this case, it's only active at the H2 receptor, and this gives you information on the type of activity, the affinity, and the references. As I mentioned, we also provide information on clinical data. So here we give a summary of the clinical use of this particular ligand, the mechanism of action and pharmacodynamic effects, and then link out where you can find information on ADME data for this particular ligand. So in addition to compounds, we also provide information on peptide ligands, in this case endothelin-1. So we provide curated sequence information, 1-letter and 3-letter amino acid sequences, as well as post-translational modification of the ligand and modification of chemical groups. We provide information on the precursor protein and the encoding gene. This is in the case of endogenous ligands. And there's also information on similar sequences. So you can search through sequences to find peptides with similar sequences in the database. For many of our targets, we also provide customized tables. And so these are just listed down here. I won't go through that list except to show you some of these. So, for example, for heteromeric receptor complexes, in this case the GABA-B receptor, we provide information on the different subunits that are associated with the GABA-B complex. Selected 3D selected 3D structures with links to PDB are provided. And then in the case of ion channels, gating inhibitors, channel blockers. For nuclear hormone receptors, information on DNA binding, so the actual sequence that the receptor binds to, co-binding partners, the main co-regulators and the main target genes for a particular nuclear hormone receptor. And then in the case of enzymes, we provide information on substrates and reaction kinetics, cofactors and inhibitors. And for example, for transporters, we also provide customized tables. So I touched on the search functionality of the database earlier. So there's two ways you can search the database. The first is through the quick search function at the top, which provides an autocomplete function. So as you start typing, you'll all be aware of this. It'll give you information about either targets, the families, or the ligands that contain the words histamine in them, for example. And then there's also the advanced search functionality in the database. So advanced search function for targets. You can search by name, a keyword, identifier, for example, UniProt accession, or references. So these can all be selected for over here, and then it will return research results based on the criteria that you've selected in the advanced search box. Similarly, for ligand search tools, there is an advanced search functionality there. I mentioned that you can search through identifiers. You can also search through substructures. So, for example, you can edit a ligand structure in this editing tool over here, and then ask the database to return results of similar structures of ligands in the database. You can also search by keywords in the database. So, for example, if you input a search with the words Alzheimer's disease in it, it will return all targets and ligands that have been implicated in Alzheimer's disease that we have in the database. And we're working this up for diseases in total at the moment. Right, so just recently we started adding information on antibodies, kinases, proteases, and also epigenetic targets into the database. So we have a collaboration with the International Immunogenetics Information System, IMGT, and we provide data on 67 approved and experimental therapeutic monoclonal antibodies. So this is a page for golimumab, for example, and its biological activity and clinical data. For kinases, we've created database pages for all the human protein kinases and selected lipid kinases. And we also provide detailed annotation on 30 clinically used kinase inhibitors including the affinities, clinical use, and ADME data, and selected bioactivity data for approved kinase inhibitors. And we've classified all the kinases in the database according to a fairly well-known classification system in order to organize the kinases. And in addition to that, more recently, through collaborations with Discoverix, EMD Millipore, and Reaction Biology, we've also introduced published screening assays for kinase inhibitors. And we also now provide links to the Discoverix tree spot compound visualization, profile visualization tool. So this is just an example of two of those tree spot images. So this circle represents the whole kinome, and the spot indicates the selectivity of the particular inhibitor across the kinome. So you can see that imatinib is a lot more selective across the kinome than this inhibitor over here, which shows rather poor selectivity. As I mentioned, we also provide information on proteases, so 175 proteases and 14 hydrolases that have activity records in Chembl, along with detailed ligand activity mappings for 46 of those proteases and 14 hydrolases with either approved prodrugs, drugs, or clinical candidates. And we've classified them in the database according to the MIROPS classification system, which is shown down here. And this is just an example of the presenilin-1 inhibitor page for all the inhibitors that we now have in the database of presenilin-1. And then epigenetics, we've also just recently started including those targets in the database, including chromatin-modifying enzymes and bromodomain-containing proteins, along with activity data for published inhibitors. And this is the sort of family tree that we have for those. Just recently, we've had an IUPHAR review on epigenetic pathway targets, which we've now included all the data for those inhibitors in the database. Right, so I just want to change track just slightly here and tell you about the Concise Guide to Pharmacology, which is a publication that we produce in collaboration with the British Pharmacological Society. So essentially, the Concise Guide to Pharmacology replaces GRAC, or the Guide to Receptors and Channels, and it's still going to be published once every 2 years as a supplement of the British Journal of Pharmacology. And what it is essentially is a snapshot in time of the concise target family summaries of in the Guide to Pharmacology. And it will be published as a series of PDFs and is intended to be a quick desktop reference guide. And so the first version of the Concise Guide to Pharmacology was the 2013-14 publication, and we're gearing up now for the next one, which will be the 2015-2016 version. And the Concise Guide to Pharmacology is published directly from the database. So this allows the publishers to include all the embedded hyperlinks in the PDF files. So all the gene names and the UniProt IDs link to the corresponding pages on the HGNC website or the UniProt website. And all the target and ligand names link directly to the corresponding entries in the Guide to Pharmacology. So from the PDF, you can go and get more information about the target and the ligand. And likewise, all the PubMed IDs link directly to the citations in PubMed. And the Concise Guide to Pharmacology can be downloaded free from the website or from the publisher's website. And this is what the front page of the Concise Guide to Pharmacology looks like. And you can see that the PDF looks very similar to what the GRAC publication did, except it's now arranged in landscape format. And although I can't show you on this particular slide over here, but every single one of these identifiers here links through from the PDF to the corresponding entries on HGNC or PubMed. The ligand names link directly to the Guide to Pharmacology, and the PubMed IDs link directly to PubMed. Something else I just want to make you aware of, many of you may be aware of it already, but we've been collaborating with the British Journal of Pharmacology since August 2014 to include a table of links in all published BJP articles that includes both review articles and research papers. And these tables of links provide links from all the targets mentioned in the manuscript and all the ligands mentioned in the manuscript directly through to the Guide to Pharmacology website. And we're in discussion with other journals to implement this sort of functionality as well. Okay, so this is just a close-up of what the target and ligand tables look like in BJP. Right, so additional features and resources of the website. Like many other websites, we provide frequently asked questions and help pages. There's a tutorial and a website video walkthrough of the website. Information on NC-IUPHAR nomenclature guidelines, full publication lists for NC-IUPHAR, hot topics in pharmacology and latest receptor ligand pairings for orphan GPCRs. If you wish, you can download all the data in our database. And we also provide target and ligand lists extracted from the literature as supplementary data to large reviews on drug targets. And we also have a blog post which details curation and technical aspects of the database. We publish a newsletter twice a year, which you can download from the database, but you can also sign up to receive email alerts about new content and news items. You can follow us on social media websites, and you can download all our slides and posters via SlideShare. And you can also email us. We love to get emails from visitors to the database, particularly when they have suggestions for how we can enhance the content in the database or perhaps improve content, or if someone spots a mistake, which can happen. And then just to finish off, I've very kindly been provided with a booth in the exhibition in the events hall here, and I'd be more than happy for you to come along and have a chat with me. I can take you through navigating the database if you so wish, and answer any questions that you might have. So finally, I'd just like to acknowledge the late Professor Tony Harmer, who was the founder and original principal investigator for the database, members of NC-IUPHAR and the subcommittees and Concise Guide to Pharmacology contributors, the database team in Edinburgh, and then our funders, IUPHAR, BPS, and the Wellcome Trust. So thank you very much for your attention.